key finding of our study is the identification of a new mechanism by which a type of cancer called B-cell lymphoma can happen. We have been studying a specific type of lymphoma called malt lymphoma. And in a subset of malt lymphomas, the cancer cells actually possess what's called a fusion protein. This fusion protein is abnormal, and it consists of one part of one protein fused to another part, and the two parts are called the API2 and the MALT1. And when these two parts are fused, they actually take on a new and dangerous activity. What happens is the API2 part of the fusion attracts an enzyme called Nic. And when Nic is attracted to the fusion, the MALT1 part of the fusion then cuts Nic into two pieces. You might think that cutting Nic into two pieces destroys Nic, but instead, cutting Nic into two pieces actually makes Nic super powerful. What happens is when Nic is actually cut by this fusion protein, the cutting removes what we call a regulatory portion. And when the regulatory portion of Nic is removed, then its activity is let loose in the cell, and it acts somewhat like a renegade. And when you have excessive, unchecked Nic in a cell, that actually promotes the growth and the proliferation of the cell.
So Nic is what's known as a kinase. It's a type of enzyme that's capable of altering the function and properties of other proteins in the cell. And that can have a number of different consequences. One of the consequences for certain kinases, in particular Nic, is the ability to affect whether a cell lives or dies, whether it multiplies, whether it survives and grows. And one of the reasons that this is especially exciting to us is that two activities here, the ability of Nic as an enzyme to act as a kinase within the cell, and the ability of the API2 MALT1 fusion protein to cleave Nic into two pieces, are both activities that are considered by the pharmaceutical industry as druggable. That is, those are two types of activities that historically the pharmaceutical industry has been successful in developing drugs to block those types of activities. So we think that we now have two sites to go after in terms of developing pharmaceuticals to combat malt lymphoma. We're very optimistic that eventually we could, in the pharmaceutical industry, could work to develop drugs that could target those activities. We're not there yet, but we have something to go after.